Today we're going to talk about aspects, like I said, of some details with antibody function, but also some details related to antibody diversity. And if you recall, last time I told you about our 5 antibody isotypes, IgM, IgD, IgG, IgE, and IgA, which vary based on their heavy chain constant regions. As Brian correctly pointed out, there is also a constant region on the light chain. There are 2 versions of it called kappa and lambda. Today I'm going to be telling you about some of the differences in the heavy chain constant region in terms of function. So why IgM does different things than IgE, for example. As far as kappa and lambda are concerned, in terms of those different constant regions of the light chain, there's absolutely no difference between them. And so we really don't reference that particularly frequently. A big part of what I want to tell you about today is sort of what's unique about each of these isotypes. It ties together some of the things we talked about on Friday. One other piece of information that I want to tell you now that we will come back to later and see much more information about is that the antibody protein that is made during a response can change over time. One change that happens over time is that the antibody will actually have a change in its affinity for antigen. So you can see that our antibody will go from binding kind of okay to binding really well for a particular antigen. That's listed here as something called affinity maturation. But more importantly, for what we're talking about right now, the antibody can also switch which constant region is made. And so we can have at first, in a response, an antibody that recognizes a particular microbe, a particular bacterium that is an IgM antibody. And as the response goes on, antibodies binding to that same microbe might now have a different isotype, a different constant region. Like here you can see we've seen a switch to IgG. That's known as class switch. When we get into some of the kind of molecular details of how some of this works at the level of the gene encoding the antibody, I will talk more about mechanistically how these things happen. Right now, I just want you to know that, yes, it does happen. And the reason why I want you to know that is as I'm going to tell you about different isotypes, you will see that different isotypes are good in different situations. And so it is actually possible to switch isotypes to have the isotype made that is the appropriate one for that situation. I told you before that I like to use the mnemonic MDs give everyone apples. And again, this is a little bit of oversimplifying it for the moment. But if I were to think about timing in a response, the first one that's made is M. And then later on, you see some of these other ones made. And so that's one of the other reasons I like this to remember the order is because MDs give everyone apples tells me, oh, right, M was the first one. And this might be some of the ordering on subsequent ones. So here you can see some of the unique aspects of some of these different isotypes. I've got 2 slides that are 2 different figures from your textbook. This is figure 4.29. The next slide is going to be 4.30. And so they are 2 parts of your textbook that are looking at the unique aspects of isotypes. One thing that this table as well as the others do, is it does break down some of the isotypes into subtypes. So you can see this doesn't just say IgG. It tells you that there are multiple kinds of IgG. I'm not going to get like super into that. Similarly, there's multiple types of IgA. You just know about IgG. That's fine. But what you can notice if you just look at the aspects of isotypes is that some of them might vary in molecular mass. Some of them will vary in level in the serum. Some of them vary in terms of half-life. You can also see from this other table that they vary in terms of what function they do. So on Friday, I told you about some of the functions, the effector functions of antibodies. All of the isotypes don't do all of those effector functions. There's some link between which isotype we have and which effector function we have. And so that's another place where those isotypes are unique. And so I'm going to kind of walk us through what each of these isotypes do, what's important about them. So we can first start talking about IgM. And I've got a little red box around IgM on this table. So what do you notice really stands out about IgM from this table? What's unique about IgM? Yeah, Brooke? It has a large mass. You can see that the molecular mass of IgM is way bigger than all of the other ones. And the reason for that is that when we find IgM in the body, it doesn't look like this. Instead, IgM is usually found as a pentamer. And so we have 5 copies of the same IgM molecule attached together. So there's this little joining protein, the J chain, that actually brings 5 IgM molecules together. And you can actually see some images of pentameric IgM from an electron microscope here. And so that's kind of one of the key things about IgM compared to the others is that IgM is usually found as a pentamer, while others are not usually found as a pentamer. And so that's pretty nice for the IgM molecule because now it doesn't have 2 identical binding sites for antigen. It has 10 identical binding sites for antigen on this molecule. And so that can really help it bind particularly well. As you will see later on, IgM is made really, well, as I've already told you, and I will tell you again, IgM is made really early in the response. That may be before we've had that change in binding strength. So it might be a little bit of a weak binder, whereas when we have the change over time, we're going to get better binding. And so IgM has helped out that, yeah, maybe it has a little bit of weaker binding, but it's got 10 binding sites instead of only having 2. So that really helps it. When I think about IgM, in addition to, as I have mentioned, being made early in the response, IgM is always made first. There's one other thing that stands out to me as a key feature of IgM. And you can look at this slide in this table and see what is the thing about IgM as well. Yeah, Jonah. IgM is very good at activating complement, and that's mainly the thing it's good for. And so IgM is really good at activating complement. Yeah, there's some other IgGs that activate complement, but if I wanted to activate complement, I always think of M. I never even think of these Gs. I'm always like, yeah, IgM is my complement activator. You can see it does a couple other things sort of meh, but really it's the star of activating complement. The reason why IgM is such a star at activating complement is because it makes that pentameric structure that I showed you, where it has 10 binding sites for antigen, but also 5 FC portions. This structure, geometrically, is a perfect match for C1. And so, yeah, like other antibodies could turn on C1 if they happen to bind at just the right angles. But IgM has the perfect structure to bind C1. And so IgM, particularly pentameric IgM, is perfect for starting the classical pathway of complement. So those are the fun facts about IgM. I'm not going to go in MD's give everyone apples order here. There's a reason for that. I'm instead going to switch us to IgE first. And I'll be honest with you, one of the reasons why I switch us to IgE at this point is because it will make my life easier when I tell you about IgD. So if you look at this table about IgE, what do you notice about IgE? Yeah, Brenna.
It has the highest serum level, but the shortest half-life, or second shortest half-life.
So it has a pretty short half-life. And what's going on with the serum level?
Small.
Has by far the smallest serum level. So if I draw some blood and take some serum, I'm probably not going to find any IgE. You have very little IgE in your serum. And its presence in the serum is on the short side. There is a reason why IgE is found so rarely in the serum. It turns out it's not because you make a tiny, tiny amount of IgE. So if it's not that you make a tiny amount of IgE, what could be the reason why there's not so much IgE in the serum? Yeah?
You are making enough, but it's being used.
Okay, it's being used. There's even a simpler way of phrasing it than that. It's somewhere else. It's somewhere other than serum, right? And that is true. And you're going to see that as I talk about effector function. So IgE is not found free in the serum. It is found somewhere else. When you look at this table and think about IgE, what do you see as sort of the unique standout feature of IgE? Yeah, David.
It activates other immune cells, specifically mast and basophils.
Yeah, it really is key for activating mast cells. And so IgE is the one that activates mast cells. When I told you about mast cell activation on Friday, I told you that mast cells had an Fc receptor on their surface, and that Fc receptor could bind to the Fc portion of antibodies. Well, it turns out it's not just any old Fc receptor. It is an Fc epsilon receptor, so it specifically binds IgEs and not all the antibodies. And so when I think about IgE, I really think about activation of mast cells. And so I need to give you a little more info about what's going on in this figure with activation of mast cells to fully explain IgE. Because if you recall, like 2 minutes ago, I told you that we don't see a lot of IgE in the serum. It turns out as soon as IgE is made, and as soon as it is in the blood, it immediately binds to an Fc receptor on a mast cell. So all of your IgE that you have right now is all stuck to mast cells. It's not free in the serum. And those mast cells might look something like this, full of all sorts of granules, and you can't really see all the IgE on their surface. If it happens that the mast cell comes in contact with the antigen that IgE could bind, the antigen will immediately bind to that antibody, which is already bound to this Fc receptor, and that will make the mast cell release all of its granules. And so here you can see a mast cell already has IgE on its surface. It's just floating around your body with the IgE already on its surface. If you happen to come in contact with the antigen, now the mast cell is going to immediately release its granules at that microbe to try to kill that microbe. And so IgE is sort of unique, again, because it is all found in the surface of mast cells, having bound to the Fc epsilon receptor on the surface of mast cells. It's not free floating in the blood. And really what your IgE is doing is activating mast cells. That's particularly good for dealing with responses to parasites like the helminth that is shown here, or if you're me, to all the things you're allergic to. So in talking about immunoglobulin isotypes, I've told you about IgM and IgE, and now I want to tell you about IgD. When I show you the table that I've been showing you about different antibody isotypes, I'm going to start with a version of the table from an old edition of the textbook. So I'm going to show you an old table about IgD before I show you the new table. So this is the old table about IgD. This is not the old table that I want it to be, but that's okay. If I had the old table that I'm used to looking at for IgD, there would be a minus sign basically for everything. When I first learned about IgD, what I learned about IgD was that it had no function, or at least no known function. When I first learned about IgD, I learned, oh, it's an evolutionary relic that sort of stuck around that doesn't do anything. And the only reason that I ever think about IgD is that B cells make IgD during one particular phase of their life. And so if I, as an immunologist, wanted to know what phase of life a B cell was at, I could look and see if it had IgD. And that would tell me that the B cell was at this stage of its life and not one of these other life stages. And that is literally the only reason I really ever think about IgD. Now obviously seems a little weird, like we did not evolve a protein to help make our flow cytometry easier. There's something going on with evolution there, but we're not really seeing a whole lot of what's going on with IgD. There are a few disease types where we have observed that patients have excess IgD, but why we care, we don't really know. Though in more recent years, we have been able to come up with a potential function for IgD. And this function also, if we think about it, kind of helps us think about why we were having so much trouble understanding what's up with IgD. So if you look, what we now think is that IgD sensitizes basophils and acts on basophils in sort of the same way that IgE is acting on mast cells. And so we really think of IgD as sensitizing basophils or activating basophils. Why do you think that that would be something that's hard to study and that we don't understand super well? Yeah. There's really few basophils, particularly in the blood. And so if we can't find very many basophils to study, then it's going to be hard to do an experiment to see that, oh, look, this antibody is bound to the basophil that we can't find. And so this may be sort of a limitation. Maybe they're doing something really important, but we haven't been able to fully understand it because we can't find enough basophils. And only in sort of more recent years have we found enough basophils to make this work and to be able to pull off this experiment. So that's IgD. Then we can think about IgA. So one thing that you can notice about IgA is that in a lot of these features, it's kind of average. The one thing that I find a little bit weird about this, although if one of my podcast co-hosts was here, she actually works on IgA a lot, she would yell at me for underestimating IgA, is that I often think about IgA in one anatomic location. And so when I see this, I'm like, it shouldn't be in the serum. There's not that much in the serum. It's somewhere else. And she'd be like, no, it's in the serum too. Yes, it's in your other anatomic location, but it's also in the serum. Don't forget it's in the serum. And I'd be like, no, it's in the other location. So I can already hear Steph yelling at me right now. But so one of the things, as you'll see, is what's going on with anatomic location. And that tends to be something we think a lot about with IgA. Most every immunologist who you will ask will tell you about IgA and anatomic location as being really key. But as my friend Steph would tell us, it's not just there. It does other things. People should think about it. And she does. So you can see that IgA is really good at neutralization. Yay. But IgA also, you can see, is important and sort of stands out because of its anatomic locations, which are shown here at the bottom. And IgA can sometimes be found. Again, if you asked me, I would be like, this is how IgA always is. And Steph would be like, sometimes, not always. As something called a dimer, something called secretory IgA, which is formed as a dimer. And so here you can see we've got two IgA molecules held together by another protein called the J chain. You can see some secretory IgA, some dimerized IgA under an electron microscope. So, like I've mentioned a couple of times, IgA is really important in its anatomic location. And so I kind of want to draw something, and I kind of know that I don't have the artistic skills for this drawing. So this will be fun. Let's see here. Okay, there's a person. So this person has many internal organs, one of which is a GI tract. Okay. So we also, in this person, have blood in various places. I am not going to draw the blood vessels. And we have B cells in those blood vessels. And the B cells are secreting antibodies in the blood, right? There's my antibodies in the blood. Let's imagine that I ate some kind of contaminated food. Where's the contaminated food in my excellent drawing here? In the GI tract. It's like here, right? Can my antibody access that? No, there's a wall in the way. There's a wall of cells in the way of that space. That's good because that wall is going to help keep the microbe out of the rest of the body, but it's also going to keep the antibodies from being able to protect me in this space because there's this wall in the way. And that's true of the GI tract. That's also true of the lungs. So if I wanted to have an antibody in the lungs, there would be like a cell barrier in the way. That would be true of the genital urinary tract. That would be true of all of your mucosal sites, many of your barrier organs. It's like the point of having a barrier, right? IgA can get carried across those barrier cell layers and be put into those mucosal sites. So IgA has a specialized receptor that it can bind to when it's made in the blood, and it can basically get put inside of those cells that make up that barrier layer of, say, the GI tract, and get transported across that cell layer so that you have IgA in your GI tract. You don't have any other antibody isotypes in your GI tract, but you have IgA. So before the microbe even gets out of your intestine, IgA could potentially neutralize it. This process of getting the IgA across the cell layer is called transcytosis. You can see this here as well. So you might have a situation where we got this bacteria that is in the GI tract. So here we've got this internal part of the GI tract, the GI lumen, with bacteria. The blood is over here. So that's where the antibodies would be. And you could imagine the bacteria could grow a lot and make you really sick. But fortunately, we have IgA that can cross this cell barrier. And so there's IgA in this location. There's also IgA in your lungs. There's IgA in your genital urinary tract. There's IgA in your eyes. And that IgA can neutralize the microbe before it even really kind of further invades your body. Someone sent me a mug this weekend. The mug says Immunology Student Tears on it. And if I really had a mug full of Immunology Student Tears, because they are tears, they would have lots of IgA in them. And so here is, you can see sort of this key piece, which has to do with anatomic locations of IgA. And so IgA is shown in yellow, what I think of as IgA. That is technically just IgA that's in a dimer. You can see that IgA that's in a monomer can be found in lots of places. And that's the one that I often forget about. And my friend Steph would be like, remember that one exists too. I don't usually think about that one. I mostly think about dimeric IgA. And what you'll notice is that dimeric IgA is in the lung, it's in the GI tract, it's in the genital tract, it's in the eyes, it's in the nose, it's in the mouth. You can see all of those sort of body locations where IgA is particularly good at being able to neutralize microbes. There's one other location where IgA is found. And this is also due to its ability to cross particular types of cell layers. IgA is also found in breast milk. And so it is really the only isotype that's found in breast milk. And so when a baby is breastfed, that baby is actually getting IgA from its mom. And so you can see this baby getting IgA from its mom. So there's one other thing that people often find really interesting about IgA. Throughout this semester, we will see some examples of immunodeficiencies, which are situations where you have too little of some aspect of your immune system, where you maybe have a mutation so that some part of your immune system is missing. Perhaps you're more susceptible to microbial infection. The most common immunodeficiency in humans is something called selective IgA deficiency. You can see numbers depicting frequency of selective IgA deficiency in this table from your textbook. So this is actually incidence per million individuals. You can see that depending on the population group we're looking at, the frequency varies a bit, but it's pretty frequent. And what is sort of interesting about selective IgA deficiency is that it generally seems like people who have this are entirely healthy. So you could have selective IgA deficiency right now and not know it. We often don't see any impact of selective IgA deficiency. One idea that has been hypothesized is that that's really because of modern hygiene, and perhaps in the past it was a bigger problem, but we can't really give you great evidence on that. When people have tried to do really large scale epidemiological studies, they say, well, maybe there's more chronic lung disease in people with this deficiency in industrialized countries, and maybe that's due to like air pollution. Maybe there's differences in celiac, but none of it is really clear and obvious. By and large, if you don't have this one isotype, if you're completely missing this isotype, you seem to be pretty much okay. And sometimes people will look at this and be like, this seems really weird. What's up with this? And why is it that we haven't had, evolution select for, getting rid of this in the human population? Why haven't we eliminated this? And one idea is because perhaps the people who would have the biggest problem here are children of IgA deficient mothers. So let's imagine if this baby has selective IgA deficiency. This baby has that disease, but its mom does not. Mom is normal. Baby has the disease. What's going to happen to the baby? Yeah. Yeah, it's going to get IgA from its mom, and so it's going to be okay. And so when it's really susceptible as a baby, it's going to be fine because it was getting IgA from its mom. What if mom has IgA deficiency? Does that mean that baby definitely will? Because of genetics? Is baby going to be genetically identical to mom? No. So if mom has it, baby might not. Mom might not give any IgA to her baby, but the baby's going to have an okay immune system to take care of everything else. And we know, while yes, this is a super complicated field of biology that I am not going to deal with right now, though it is fascinating. Yes, you can, live if you were not breastfed as a child. You are not going to die. So you know that IgA, that not having that IgA isn't going to kill you. So you can sort of see that maybe there hasn't been a lot of selective pressure because the people who might be most susceptible are the babies who wouldn't be the ones who actually had the gene, it would be their moms. So this ends up leading to sort of some complicated arguments. But what people often find really interesting is that it is entirely possible that Certainly someone on this campus at least has IgA deficiency and doesn't know. And then our final isotype is IgG. So I need to tell you a few things about IgG. You can notice there are, in truth, multiple subtypes of IgG, though I'm not going to really focus on that IgG a ton in terms of like distinguishing between the different types. But if you try to look at IgG, where do you see things that kind of stand out? Yeah, Brian. So what's up with serum level? It's pretty high in the serum. So you got a lot of IgG in the serum. And then what's up with the half-life? They last a long time. So IgG is the one that you have by far the most of in the serum. And IgG tends to hang out and persist in the serum for a long time. Brian pointed out that there is some variation in that serum level and in the half-life between the subtypes. The reason for that is actually because the biggest place they vary is in the hinge region. And this one, IgG3, has a super long hinge that can get cut up really easily by proteases. So this one can get damaged really easily, whereas some of the other ones can't. And so this one you have way less of. And this one lasts much less time in serum. So if I go back to where I just was, you can see that's the one that has the really low serum level, and that's the one that has the really low half-life because it can be degraded so easily. IgG is great at pretty much all of the functions. Yay, IgG. You can see IgG is a really good neutralizer. It's a really good opsonizer. It is, and in fact, it's sort of the main opsonizer. It is the only one that's going to do NK activation. It can activate complement. So really, IgG can kind of do a lot of our functions. Again, when I think about IgG, there are a couple of big things that I think of. One of them has to do with the timing that it is made in the response. So I've already told you about IgM, which is shown in black, being made kind of first in a response, and other types being made later, and IgG is shown in red. And so you can see IgG is made a bit later than IgM. If this was a fully complete image, where it shows IgM being made time-wise, it would also, IgG being made time-wise, it would also show IgG and IgE. So M and D are kind of M, then D, then G, A, and E. Or G, E, and A. But and you can also see that if we look at a secondary memory response, we make tons of IgG. And so if you see that a patient is making IgG, you know they're either kind of late in their primary response, or especially if they're making a lot of it, you know they're making a secondary response. And again, you would, time-wise, this would also be IgE and IgA. The issue is, if you recall, you don't really have very much IgG and IgA, or IgE, E and A in the serum. And so if you were going to be the physician who wants to do this measurement, you'd be like, it's a pain in the butt to measure IgE. I'm going to measure IgG because it's really a lot. And so we tend to do IgG measurements to tell us about sort of the maturity and quality of an immune response, to know that it's sort of a good secondary response. We could theoretically do A or E time-wise, but there's just so much more G. And so G is the one we measure a lot when we want to characterize immune responses. So if, for example, I wanted to go to the doctor and say, hey, I want to see if like my measles vaccine that I got when I was a little kid still works, they would actually draw some blood and see if I have any measles IgG. And so IgG is sort of a key one that we do a lot of measurements of. Just like with IgA, IgG also can have a unique anatomic location. It's not its main anatomic location, but it's the only one that can go to this place. And that is across the placenta. So in most people, IgG is all through the blood and you've got lots of IgG. In the case where we have a pregnant woman, there is also IgG in the placenta, and IgG is the only one that can be in the placenta, and thus IgG is transferred to the fetus. And so that's part of the reason why that IgA in the breast milk, may be a little redundant, because that fetus doesn't just have IgA that it got from its mom, it also got some IgG through the placenta. So IgG is the only one that is transferred through the placenta. This happens kind of similarly to the IgA process in that there is a receptor that is helping carry the antibody across a cell layer. In this case, it's a cell layer which is the placenta. You can see this in the trophoblast that's separating the mom's blood and the baby's blood. And we've got this special process to transport the antibody across without that antibody getting destroyed. So what you can notice here is actually the details of antibodies in fetuses and in infants. And so if we look at birth here, at birth, we have a lot of IgG that came from mom. And the baby actually hasn't really started making a bit of IgM on its own, but it really hasn't been making anything else. And so early on, the baby is sort of perhaps uniquely vulnerable because it just has IgG from mom. Mom doesn't give it any B cells, so it can't make more of that. It just has some of that. And in fact, you can see it goes away pretty quickly because it's just a protein that was transferred. That is going to eventually degrade. There are no B cells there. And you can see that there's this period of time where eventually the baby's immune system will start making some of these antibody types themselves. But you can see that sort of there is this window of time where the baby might be particularly susceptible because it has relatively low levels of some of these antibodies. This information is particularly important for people who have designed vaccine schedules to think about when to give babies a vaccine. If you give babies vaccines too early, then this IgG actually will destroy it before the baby's B cells have time to do something. But if you wait too long, then you let the baby spend all this time being super vulnerable. And so we've spent a lot of time designing vaccine schedules so that we know, well, when is the baby's immune system going to be able to recognize, to work, and can we make sure we're giving vaccines right there to keep that baby from being vulnerable for a long period of time. One other big thing that people have been thinking about a lot on this front is that, okay, we know that IgG from mom can cross the placenta. So what happens if we give vaccines and we immunize pregnant women? Can we actually protect their babies by having those antibodies go across the placenta and go into the fetus? And there's lots of great evidence on that. One of the most famous places where there's evidence on this is that pertussis, whooping cough, is really deadly to really small babies. They really need to be protected from pertussis. And so the CDC has actually changed recommendations so that pregnant moms now get a pertussis vaccine in their 3rd trimester, with the idea of making a bunch of pertussis antibodies to passively transfer to the baby across the fetus or across the placenta, so that the baby is protected. And there has been discussion about many other vaccines and whether getting them in certain parts of pregnancy might confer some protection to the baby because those antibodies are passed through the placenta. Usually, this involves getting those vaccines either in the 2nd or 3rd trimester. One other, and this really is an example of passive antibody transfer. I told you also about passive antibody transfer as a treatment in some situations. There is also one other sort of passive antibody transfer situation that I will mention, which is that there are some autoimmune diseases that are driven by antibodies. If a mom has an autoimmune disease driven by antibodies, like in this case, Graves' disease, that impacts the thyroid, those antibodies can cross the placenta and impact the baby, and the baby can be born with some symptoms. The good news in this case is that the baby didn't get any B cells from mom, just those antibody proteins that have crossed the placenta. So those antibodies are not going to last the baby's whole life. They will eventually degrade, as all proteins do, and the baby will get better. A. And B, we know how to prevent this now. And so we can actually protect this baby with some other medications that we have. And so this doesn't end up being as big of a deal. Yes, Brooke.
Is it passive immunity from the mom, so you can only give like vaccines that have IgG?
So the vaccine itself doesn't have IgG in it, but the vaccine makes your immune system make IgG. So you could only get, so you're exactly correct that if we had a vaccine that made only IgE or something, that would not work for passive antibody transfer. It would have to be a vaccine that induced IgG, because IgG is the only thing that's going to get transferred. You might imagine, well, could I give a vaccine that gives you IgA and have that go through breastfeeding or something like that? But IgG is sort of what you're really trying to do here. Great question. All right, so basically what I'm learning right now is that I am pretty much exactly a whole day behind, because this is where I wanted to start today. So I'm just going to tell you what the problem is, and then we'll see what happens on Wednesday. It is possible that right now I've posted a paper to discuss on Friday. We're going to see how things happen with that. We're going to say that's tentative, and the biggest reason for that is that if you notice there's a VDJ problem set due next week, I want you to make sure you have the information for that problem set in time. And I want you to have that problem set in time to meet with me and also to kind of be studying it as you get ready for the next exam. So I definitely want to make sure I get through that material this week. So what happens with the paper on Friday? We'll kind of see how Wednesday goes. But let me just give you the problem that underlies all of this. Landsteiner showed us this massive diversity of antibodies. How many antibodies you can make, even against things that don't even exist yet, like these animals were making against these dyes that had never existed before. And if we actually look at how many different antibodies against how many different epitopes you have, it's estimated that we can make about 10 to the 16th different antibodies. Right? That's why I'm getting confused. 9, 12. Do I need 2 more? Okay. So then it's that many of these different proteins. David, I have a very challenging question for you. Where do proteins come from? you got to transcribe a RNA, right? And you had to have a gene. So you should have a gene to match each protein. When we look at the human genome, that's how many genes you have. So there's a bit of a problem here in terms of how do we use this many genes to make that many proteins. And even I, who loves immunology very much, would say you can't have every gene in the entire body be used for making antibodies. You probably need a couple other genes for doing other things. So there is a big math problem here. And so we're going to have a really sort of unique genetic process to make these proteins. And that's what we're going to start talking about on Wednesday. And so I'll see you guys on Wednesday.